When similar kind of cells work together, we call it as tissue. Tissues are of different types depending on the functions, structures, types of cells, etc. But when several tissues are grouped based on the structure and location, they form the tissue system. In this session, we will discuss the different tissue systems seen in plants. In plants, based on the structure and location, basically there are three tissue systems. The epidermal tissue system towards the periphery of the plant body. Ground tissue system is between the epidermal and the vascular tissue systems, and the last tissue system is the vascular tissue system, which is towards the center of the plant body. Let's start with the epidermal tissue system. So, can you name the tissues constituting the epidermal tissue system? It is epidermal cells, stomata, and many epidermal appendages. Epidermis is the outermost layer in tender parts of the plant body. If you look at the epidermal cells, they are made up of elongated, compactly packed cells forming a continuous layer. Well, which types of cells makes the epidermal layer? The epidermal cells are single-layered with thin cell walls. They have large vacuoles with a prominent nucleus. As the vacuoles and nucleus occupies most of the space, the amount of cytoplasm is less in epidermal cells. All these cellular characteristics makes clear that epidermis is made up of parenchymatous cells. Epidermis is usually covered by a thick waxy layer called cuticle. This layer prevents the loss of water due to heat. As the roots are seen below the soil, cuticle is usually absent in roots. Also, the epidermis in leaves is not continuous and are interrupted by stomata. We will see the structure of stomata. A stomatal apparatus constitutes the stomatal aperture, the guard cells, and the surrounding subsidiary cells. Stomata is the structure that helps in the gaseous exchange and transpiration. There are two guard cells in each stomata. The guard cells are bean-shaped in dicots and dumbbell-shaped in monocots. A pore called stoma is formed by these two guard cells. The outer wall of the guard cells is thin and the inner wall are thick, as you can see in this image. The stretching of the outer thin walls when the guard cells is turgid opens the stomata. When the guard cells is flaccid, stomata remains closed. Hence, it's the guard cell that regulates the opening and closing of stomata. Guard cells also contain chloroplast. Therefore, they undergo photosynthesis. Few cells of the epidermis found near the guard cells have specialized cell shape and size and are known as subsidiary cells. Now that you are familiar with the structure of the stomata, we'll move on to the different epidermal appendages. The common epidermal appendages are root hairs and trichomes. Root hairs are unicellular and elongated. They help to increase the surface area for easy absorption of water and minerals from the soil. The epidermal hairs of stem are also known as trichomes. They are multicellular, branched or unbranched, soft or stiff. They perform many functions. They help to reduce transpiration and therefore prevent water loss. They also serve as a barrier from insects and fungal infections. They may even secrete substance that can cause irritation to herbivores. Next is the ground tissue system. Ground tissue system is usually made up of all the simple permanent tissues such as parenchyma, collenchyma and sclerenchyma. Parenchyma is present in the cortex, pericycle, pith and medullary rays, in the primary stems and roots. Collenchyma is usually seen in the hypodermis of dicot stem, whereas sclerenchyma is seen in the hypodermis of monocot stems. In leaves, mesophyll cells constituting the spongy parenchyma and the palisade cells with thin cell wall and numerous chloroplasts becomes the ground tissues. The last type of tissue system is the vascular tissue system. Vascular system is composed of complex tissues such as phloem and xylem. The vascular tissues bundle together to form a vascular bundle. These bundles can be open or closed depending on the presence and absence of cambium. If the vascular bundles possess cambium, they are open vascular bundle and can undergo secondary growth. These kind of bundles are seen in dicots. If the vascular bundles do not possess cambium, they are closed and will not show secondary growth. This kind of vascular bundles are seen in monocots. The arrangement of xylem and phloem is different in roots and shoots. In roots, the phloem and xylems are arranged in alternate manner and are called radial vascular bundles. Whereas in shoots, the phloem and xylem are seen joined together and the vascular bundle is known as conjoined. The conjoined vascular bundles have phloem towards the periphery and xylem towards the center. These vascular bundles are arranged in a way that they fall in the same radius, hence they are known as conjoined and collateral. In ferns and many monocots, we can find another type of arrangement of vascular bundle and they are concentric vascular bundle. Concentric vascular bundles are of two types, amphicribal and amphivasal. Amphicribal is also known as hadrocentric, where the xylem is surrounded by phloem, and amphivasal is also known as leptocentric, where the phloem is surrounded by the xylem. This kind of bundles is commonly seen in monocots such as Dracaena, Yucca, etc. We have understood the different types of tissue systems in plants. Do you know there is another type of vascular bundle called bicollateral vascular bundle? Try to find out what is bicollateral vascular bundle and in which plant groups are they found. Let's now summarize what we have learned.
Manorama Horizon. Learning made simple.